Welcome you to cluster differentiation video part 2. We are going to see about the CD14. The CD14 is the lipopolysaccharide sentinel. What does that mean? It acts as a sentinel for lipopolysaccharide or LPS in short. This CD14 is not a signaling receptor by itself, but it functions as a co-receptor with toll-like receptor on the macrophages. I can explain that to you bit by bit. This is the macrophage. This is the toll-like receptor which is present on the macrophages. The CD14 is the co-receptor with toll-like receptor. Together, CD14 and toll-like receptor recognize the lipid A portion of the lipopolysaccharide. This is the lipopolysaccharide, which is the toxic component of the endotoxin. This has the lipid A. The lipid A is recognized by CD14 and the toll-like receptor complex. This recognition activates the macrophages and it triggers the innate immune response against gram-negative bacteria. They will be releasing cytokines, tumor necrosis factor, interleukin-1, resulting in an extensive gram-negative septic shock. At this juncture, I would like to emphasize that CD14 lipopolysaccharide is the septic shock. It is entirely different from Staphylococcus aureus toxic shock syndrome toxin. I have a video on explaining the differences between them. CD34 is a stem cell marker. It is not lineage specific. It is expressed on all hematopoietic stem cells. Because of this, it is widely used in clinical isolation, stem cell counting, as well as in bone marrow transplantation. CD40, the B cell activation gateway. Think of CD40 as the final handshake between B cells and T helper cells. After the primary and secondary signals are checked, the CD40 receptor on the B cells must bind to the CD40 ligand on the CD4 plus T cells. This delivers the crucial T cell help, allowing B cells to differentiate into plasma cells and to undergo class switching. Without a CD40 ligand interaction, B cells remain unactivated, resulting in hyper IgM syndrome. I do have a separate video about hyper IgM syndrome. I will list it in the immunology playlist. Paroxysmal nocturnal hemoglobinuria. Here, the RBCs are vulnerable to attack by the complement. Normally, the RBCs are protected by CD55, which is a decay accelerating factor, and CD59. CD59 is a membrane inhibitor of reactive lysis. These proteins are anchored to the RBC membrane by GPI anchors. Mutations in the PIGA gene will result in a defective GPI anchor, and therefore 55 and 59 cannot attach. Complement C3 convertase, which is supposed to be regulated by CD55, doesn't happen. Similarly, for CD59, which is supposed to inhibit reactive lysis, also doesn't happen. The RBCs are now tagged for destruction. This will result in RBCs being lysed in the bloodstream, resulting in hemoglobinuria at night. That's why it is called as nocturnal. But why does it happen in the night? Because mild acidosis will activate during sleep, will activate the complement more easily. Why should mild acidosis happen during sleep? This is because in sleep there is carbon dioxide retention, minimal carbon dioxide retention, which isn't a problem normally. But in PNH, mild acidosis is an issue resulting in hemoglobinuria. You can think this like the RBCs are armored knights with the CD55 and 59 acting as a shield or an armor plate, thereby stopping the membrane attached complex from punching holes or stopping the complement activation. In PNH, the armor and the shield falls off and the complement can now attack the RBCs more freely. CD80 and CD86. They are the co-stimulatory molecules because for the stimulation of the T cells, it follows a two-signal model. So that here when you see, this is the T cell activation. Signal 1 is the MHC on antigen presenting cells binds to the T cell receptor. MHC binding to the T cell receptor. Signal 2 is the CD80 or 86 which are present on the antigen presenting cells. This binds to CD28 on the T cells. Without this second signal, the T cells will become unresponsive or allergic. This safeguard prevents unwanted activation of the T cells against its self antigens, thereby reducing autoimmunity. Once the primary and the secondary stimulus happens, then T cell activation happens in this way. CD15 and CD30 are Hodgkin's signatures. These markers define the Reed-Sternberg cells which are pathognomic for Hodgkin's lymphoma and distinguish it from other lymphomas. CD16 and CD56, they are the natural killer cell identity. CD16 is the receptor that mediates the ADCC, antibody dependent cell mediated cytotoxicity. Here the IgG antibodies coat infected or the tumor cells in the FC segment. The NK cells bind to the FC portion through CD16, thereby releasing perforins and granzymes to induce apoptosis. This is antibody dependent cytotoxicity. Because it is dependent on the antibody, still cytotoxicity happens. This is the mechanism behind therapeutic antibodies like rituximab and trastuzumab. Apart from CD16, CD56 is an adhesion marker commonly used as an NK cell identifier. CD18 adhesion integration. CD18 is the adhesion molecule. CD18 is the beta subunit of LFA1. This is LFA1 over here. So this LFA1 has a beta subunit, that is CD18. LFA1 is an integrin which is critical for the leukocyte adhesion, as you notice here, and migration during inflammation. This process goes in steps. Initially there is rolling, followed by adhesion, then diapedesis, then chemotaxis. In leukocyte adhesion deficiency, CD18 deficiency impairs the adhesion, leading to recurrent bacterial infections because the neutrophils are unable to enter through the blood vessel wall to the damaged tissue. And there is delayed wound healing. And the damaged tissue, there is absent first because there are no neutrophils. But in the bloodstream, there is elevated peripheral neutrophils. CD19 and CD20, they are the B cell signatures. CD19 is present from pre-B cells all the way to the plasma cells, while CD20 appears on mature B cells but is absent on the plasma cells. Because of its restricted expression, CD20 is a therapeutic target for drugs like rituximab, which target CD20 to deplete B cells in autoimmune disease and lymphoma. Interleukin 2. This is the interleukin 2. The interleukin 2 has a receptor alpha chain as well as the gamma. This is the alpha. CD25 is the regulatory T cell marker. It is the alpha subunit of the interleukin 2. This is the alpha, this is CD25. They are highly expressed on regulatory T cells or the TREGs. The TREG suppresses immune responses, preventing autoimmunity and maintaining homeostasis. CD25 is a high affinity interleukin 2 binding, enables TREG proliferation and sustained suppression. You can remember it like it is a peacekeeper badge. So if it is there, then there is no fighting. The T cells are not activated. We come to the end of the slide. So I want to make it a little bit interesting to you. CD21 is on the B cells is the primary entry point for Epstein-Barr virus. You can see I have drawn it as a board, as the door, CD21. It is the entry point for Epstein-Barr virus. This is expressed on mature B lymphocytes. There is a B cell. So when the Epstein-Barr virus's GP50 glycoprotein binds to CD21, enabling the viral entry and latent infection in B cells, leading to mononucleosis and potential malignant transformation of the B cells. With this, we have come to the end of the cluster differentiation part 2. As I have promised earlier, I have made it as much logical and interesting to you. If you like my videos and would love to get mentored by me, send me a message in the comments. Have a good day.